Assalamualaikum friends. I hope so that you all will be fine. Friends, in this video I am going to discuss about one of the most important topic in carbohydrates metabolism, that is TCA cycle. Friends, I will cover this topic under the heading of the introduction, steps, significance, energetics, inhibitors, regulation, amphibolic role, and anaplerotic reaction. Friends, if you are new on this channel, then kindly like, share, and subscribe to my channel. So let's start our lecture. First of all, the introduction of TCA cycle. Friends, this TCA cycle, it was described by Sir Hans Krebs, and he got Nobel Prize in 1953. So the another name of TCA cycle is Krebs cycle. Later on, this TC cycle was confirmed as citric acid cycle. It is due to the discoverer Oxton, and in 1944 Oxton suggested that TCA cycle described by Sir Hans Krebs, it is basically a citrate. So the another name of TCA cycle is citric acid cycle. So the another name of TCA cycle is Krebs cycle, and citric acid cycle. Also, this TCA cycle is also called as metabolic traffic cycle because in this cycle, lot of intermediates are formed that are used in various other metabolic pathway. Students, viewers, friends, it is just the introduction of TCA cycle. The another name of TCA cycle is Krebs cycle and citric acid cycle. Now coming toward the steps of TCA cycle. Steps. First of all, the oxaloacetic acid that contain 4 carbon, it gets condensed, it gets condensed with acetyl CoA, which is a 2 carbon atom containing compound. Acetyl CoA. And there is a removal of coenzyme A and SH, that is sulfhydryl group. It converts into citrate, 6 carbon containing compound, with the help of an enzyme that is citrate synthase. There is condensation of oxaloacetic acid and acetyl CoA. Acetyl CoA, it is a 2 carbon atom containing compound. And there is a removal of coenzyme A sulfhydryl group, and there is a formation of citrate, which is a 6 carbon atom containing compound, with the help of this enzyme citrate synthase. In next reaction, it converts into cis aconitate, and ultimately it converts into isocitrate, and with the 6 carbon atom containing compound. And here is a dehydration reaction. Dehydration with removal of water molecule, and here is a rehydration. This process occurs with the help of aconitase enzyme. Okay. This citrate converts into the cis-aconitate by dehydration reaction. Cis-aconitate it converts into isocitrate with the help of rehydration reaction, and this process occurs with the help of aconitase enzyme. In next reaction, by action of isocitrate dehydrogenase enzyme, okay, it converts into alpha ketoglutarate, alpha ketoglutarate, okay, and there is a removal of carbon in the form of carbon dioxide, and here NAD plus it converts into NADH and H plus molecule. Okay, the isocitrate with the help of isocitrate dehydrogenase enzyme, there is a removal of carbon dioxide and NAD plus. It converts into NADH and H plus, NADH and H plus. Okay, and there is a formation of alpha ketoglutarate, which is a 5 carbon atom containing compound. Okay. In next reaction, in next reaction, this alpha ketoglutarate with the help of alpha ketoglutarate dehydrogenase enzyme, alpha ketoglutarate dehydrogenase enzyme, and there is a formation of succinyl CoA with 4 carbon atom containing compound. Basically here the carbon it gets released in the form of carbon dioxide, and CoA, coenzyme A, also gets incorporated. Here there is a incorporation of coenzyme A, and there is removal of carbon dioxide, and NAD plus it converts into NADH and H plus. This alpha ketoglutarate, by action of alpha ketoglutarate dehydrogenase enzyme, it converts into succinyl CoA. There is incorporation of coenzyme A and removal of carbon dioxide, and NAD plus converts into NADH and H plus. And there is a formation of succinyl CoA with a 4 carbon atom containing compound. In next reaction, this succinyl CoA it converts into succinate and is a 4 carbon atom containing compound with the help of succinate thiokinase enzyme. Okay, and there is a formation of ATP. Students, this step is a substrate level phosphorylation step, mean synthesis of ATP from substrate without undergoing into electron transport chain. So let's see, there is a GDP and inorganic phosphate, they form GTP, and GTP ultimately it converts into ATP. Okay, this converts into ATP. So this step is a substrate level phosphorylation step, and there is a formation of succinate with the help of succinate thiokinase. This succinate it converts into, in next reaction it converts into fumarate. Again 4 carbon atom containing compound with the help of succinate dehydrogenase enzyme, and here NAD plus, it converts into NADH and H plus. In next reaction, this fumarate, it converts into malate, which is a 4 carbon atom containing compound. This oxaloacetic acid. And fumarate converts into malate with the help of fumarase enzyme. Fumarase enzyme, and there is an incorporation of water molecule. And malate, it converts into this oxaloacetic acid with the help of malate dehydrogenase enzyme. And NAD plus, and here NAD plus, it converts into NADH and H plus. Okay, so students, these all are the steps of the TCA cycle. Okay, let's see this oxaloacetic acid, which is a 4 carbon atom containing compound. By condensation with acetyl CoA, which is a 2 carbon atom containing compound, there is a removal of COSH. With the help of citrate synthase, it converts into citrate, which is a 6 carbon atom containing compound. And citrate, by dehydration reaction, it converts into cis aconitate, and by rehydration reaction it converts into isocitrate, which is a 6 carbon atom containing compound with the help of aconitase enzyme. In next reaction, isocitrate, with the help of isocitrate dehydrogenase enzyme, there is removal of carbon dioxide, and then NAD plus, it converts into NADH and H plus, and there is a formation of this alpha keto glutarate, which is a 5 carbon atom containing compound. There is a formation of alpha keto glutarate. Okay, this alpha ketoglutarate, by action of alpha ketoglutarate dehydrogenase enzyme, there is incorporation of CoA and removal of carbon dioxide. NAD plus converts into NADH and plus, and there is a formation of succinyl CoA, which is a 4 carbon atom containing compound. The succinyl CoA then converts into succinate by action of succinate thiokinase, and GTP and PI converts into GTP, and GTP ultimately converts into ATP, and is a substrate level phosphorylation step, and there is a formation of succinate with 4 carbon atom containing compound. Succinate converts into fumarate with the help of succinate dehydrogenase enzyme, and NAD plus converts into NADH and H plus. This fumarate converts into the malate with the help of fumarase enzyme. There is incorporation of water molecule, and malate converts into oxaloacetic acid with the help of malate dehydrogenase enzyme, and NAD plus converts into NADH and H plus. Okay, so this is the TCA cycle. This is the TCA cycle. You can see this is the TCA cycle. Okay. Okay, this is the TCA cycle. And now coming toward these are the steps. Total there are 8 steps. This is the step number 1, this is the step number 2, and this is the step number 3. Okay, this is the step number 4, this is the step number 5, this is step number 6, step number 8, and step number 7, and step number 8. Total there are 8 steps of TCA cycle. So now coming toward the significance of this TCA cycle. Significance of TCA cycle. Okay. Um, Now coming toward significance of TCA cycle. Friends, oxaloacetic acid, it acts as a catalyst in this cycle, and it also gets regenerates in this cycle. So the first significant point is the oxaloacetic acid, it acts as a catalyst, and also it gets regenerates in TCA cycle. This is the first significant point. The second point is it is a common oxidative pathway for the oxidation of all foodstuff. It is a common oxidative pathway for the oxidation of all foodstuff. Now, third point is in this TCA cycle there is a formation of reducing equivalent. Formation of reducing equivalent, and that are NADH and FADH2. Now, the fourth point is in TCA cycle, the one step is a substrate level phosphorylation step. The one step is substrate level phosphorylation. Fifth point is this TCA cycle, it is very important for the synthesis of non-essential amino acid, heme, purine and pyrimidine. Synthesis of non-essential amino acid, heme, purine and pyrimidine. Friends, these are the significant point of the TCA cycle, and one of the most important point, that is TCA cycle, it takes place in mitochondrial matrix. Okay. So students, significance of TCA cycle is oxaloacetic acid, it acts as a catalyst and also regenerates in TCA cycle. Second is it is a common oxidative pathway for the oxidation of all foodstuff, and it also produces reducing equivalents that are NADH and FADH2. It is the one step in TCA cycle. The one step is substrate level phosphorylation, mean synthesis of ATP from substrate without undergoing into electron transport chain, and there is also synthesis of non-essential amino acid, heme, purine, and pyrimidine. Okay. Friends, this TCA cycle takes place in mitochondrial matrix. So now coming toward the energetics of TCA cycle. Energetics of TCA cycle. In TCA cycle, there are production of three molecules of NADH and H plus. And one molecule of NADH and H plus in electron transport chain, it gives 2.5 ATP. So 3 multiply 2.5, that is 7.5 ATP. And one molecule of FADH2, it gets synthesized. So from FADH2, one molecule of FADH2 in electron transport chain, it gives 1.5 ATP. So 1.5 ATP production from FADH2, and also the one steps that take place in the TCA cycle, substrate level phosphorylation, and from substrate level phosphorylation there is synthesis of only 1 ATP. So 1 ATP. So the total amount of ATP from one molecule of acetyl it is 10 ATP. In, as we discussed earlier, that in TCA cycle there is production of three molecules of NADH and H plus, one molecule of NADH and H plus in electron transport chain, it gives 2.5 ATP. So there is production of 7.5 ATP from three molecules of NADH and H plus. One molecule of FADH2, it gives 1.5 ATP, and there is 1 ATP from the substrate level phosphorylation step. So total amount of ATP from one molecule of acetyl-CoA, that is 10 ATP. Okay. Now coming toward the inhibitors of TCA cycle. So there is these are the inhibitors of TCA cycle. This fluoroacetate, it inhibits aconitase enzyme. This arsenite, it inhibits alpha-ketoglutarate dehydrogenase enzyme, and this malonate, it inhibits the succinate dehydrogenase enzyme. So friends, now coming toward the energetics regulation of TCA cycle. Regulation of TCA cycle. It regulation, it depends upon the availability of substrate. Okay, if ADP, it is in TCA cycle, if ADP it is present in higher amount, in higher concentration, then it will activate this TCA cycle. And if ATP and NADH, they are present in higher concentration, they will inhibit the TCA cycle. Okay. The third point is succinyl CoA. It inhibits alpha-ketoglutarate dehydrogenase enzyme. The fourth point is acetyl CoA. It inhibits pyruvate dehydrogenase complex. And the fifth point is there is a allosteric regulation of citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. These all enzymes, they are regulated allosterically. Allosterically mean allosteric. The enzyme have two sides. First one is active side and other is the allosteric side. Allosteric side, basically it is the regulatory side. It depends upon the effector. If the effector, if effect is allosteric activator, then it will activate the then it will enhances the activity of it will enhance the activity of enzyme. If the allosteric effector is the inhibitor, allosteric inhibitor, then it will reduce the activity of enzyme. So these enzymes, they all are regulated allosterically. So students, this is the regulation of TCA cycle. It depends upon availability of substrate. If ADP are present in higher concentration, they will activate TCA cycle. If ATP and NADH, if they are present in higher concentration, then they will inhibit this TCA cycle. Succinyl CoA, it inhibits alpha-ketoglutarate dehydrogenase enzyme. Acetyl CoA, it inhibits pyruvate dehydrogenase complex, and these enzymes, they are allosterically regulated. This is the regulation of TCA cycle. Now coming toward the amphibolic role of TCA cycle. Amphibolic role of TCA cycle. The amphibolic means the amphibolic means the catabolic and as well as anabolic role. So let's see catabolic role. The acetyl CoA, it forms two molecules of carbon dioxide. This is a catabolic role. Catabolism means if the breakdown of larger molecule into simplest form, and anabolism means the synthesis of large molecule from the smallest molecule. Now coming toward the anabolic role, the first one is the oxaloacetic acid. It forms aspartate, and this aspartate, it involves in the synthesis of purine and pyrimidine. The adenine and guanine, they are purine, while thymine and cytosine, they are the pyrimidine. Next one is alpha ketoglutarate. It forms glutamate and gamma amino butyric acid, that is an inhibitory neurotransmitter. The third one is succinyl CoA. It is very important in synthesis of heme. The fourth one is citrate. It acts as a precursor for the synthesis of acetyl CoA, and acetyl CoA, it is very important. Acetyl CoA, it is very important in the synthesis of fatty acid and cholesterol. Okay, students, this is the amphibolic role of TCA cycle. It is a short answer question. Amphibolic means catabolic and as well as anabolic. Catabolic role is there is a conversion of acetyl CoA and there is formation of two molecules of carbon dioxide. And anabolic role includes the conversion of oxaloacetic acid to aspartate. Aspartate is very important in synthesis of purine and pyrimidine. And alpha ketoglutarate conversion into glutamate and GABA. GABA, it is a inhibitory neurotransmitter, and the full form of GABA is gamma aminobutyric acid. Succinyl CoA convert into synthesis of heme, and citrate convert into acetyl CoA, which is very important in the synthesis of fatty acid and cholesterol. So students, now coming toward the anaplerotic reaction of TCA cycle. Anaplerotic reaction. Anaplerotic reaction means the reaction that replenish the intermediates of TCA cycle. It is called as anaplerotic reaction. So let's see the examples. The reaction which replenish the intermediates of TCA cycle is called as anaplerotic reaction. Examples are the aspartate. It forms oxaloacetic acid, which is the intermediate of TCA cycle by transaminase enzyme. This glutamate, it forms alpha ketoglutarate by action of transamination reaction and deamination reaction, and also histidine, arginine and proline, they also form alpha ketoglutarate. This is the first one. Second one, now coming toward the pyruvate, it also form oxaloacetic acid with the help of pyruvate carboxylase enzyme, which requires ATP, biotin and carbon dioxide. The fourth one is pyruvate. It forms malate by action of malic enzyme. It requires NADP plus that converts into NADPH. NADP plus that converts into NADPH. Okay. Fifth one is the fifth one is succinyl CoA. The succinyl CoA, it is formed from Propionyl-CoA, propionyl-CoA, valine, threonine, methionine, and isoleucine. Okay, and the last one is fumarate. Fumarate, it is formed from phenylalanine, tyrosine, and urea cycle. They all form fumarate. Fumarate. So students, this is the anaplerotic reaction, the reaction that replenish the intermediates of TCA cycle. It is called as anaplerotic reaction. It is very important short question. Here the some important examples are the conversion of aspartic to oxaloacetic acid with the help of transaminase enzyme, the conversion of glutamate into alpha ketoglutarate with the help of transaminase and deaminase enzyme, and also histidine, arginine, proline. These are the amino acids that form alpha ketoglutarate. The conversion of pyruvate into oxaloacetic acid with help of pyruvate carboxylase enzyme, which requires ATP, biotin, and carbon dioxide, and conversion of pyruvate into malate with the help of maleic enzyme, which requires NADP plus, which converts into NADPH, and succinyl-CoA. Succinyl-CoA is formed from propionyl-CoA, valine, threonine, methionine, and isoleucine. And fumarate is formed from the phenylalanine, tyrosine, and urea cycle. And fumarate, succinyl-CoA, oxaloacetic acid, alpha ketoglutarate, oxaloacetic acid, and malate, they all are intermediates of TCA cycle. So, students and friends, it is all about TCA cycle, their introduction, steps, significance, energetics, inhibitors of TCA cycle, regulation, and amphibolic role of TCA cycle, and anaplerotic reaction. If you have any query, you may ask in comment section. And one humble request, that is, kindly uh, see this lecture again and again. Watch this lecture again and again. And if you have any query, you may ask in comment section. Keep watching and thank you.